Hello everyone, welcome to today's live broadcast, Messengers of the Mind. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We have some important announcements before we begin the presentation. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you are interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's featured speaker, Dr. Floyd Bloom. Hello, Dr. Bloom.
Good afternoon.
Floyd E. Bloom, MD, is Professor Emeritus at the Scripps Research Institute in La Jolla, California. He was Chairman Emeritus of the Department of Neuropharmacology at the Scripps Research Institute, past President of the American Association for the Advancement of Science, former President of the Society for Neuroscience and of the American College of Neuropsychopharmacology, former Editor-in-Chief of Science from 1995 to 2000, Director of Behavioral Neurobiology at the Salk Institute for Biological Studies, and Chief of the Laboratory of Neuropharmacology of the National Institute of Mental Health. Dr. Bloom recently won the RSA Lifetime Achievement Award at the Research Society on Alcoholism's annual meeting in June 2012 in San Francisco, California. I will now turn it over to Dr. Bloom for his presentation.
Well, thank you, Judy, and good afternoon to everyone. This afternoon I want to tell you the story of how neuroscientists came to accept the concept that neurons speak to each other chemically through the agents that we call neurotransmitters. Before we begin, I should disclose that I'm a founder and director of Alkermes, a company based in Dublin, Ireland, and a director of two other small companies, but I won't mention any of their products this afternoon. So you may be wondering, and if you're not wondering, perhaps your students will wonder, why should they study the history of neuroscience? Well, my concept is based on a book I recently read called How We Got to Now by Stephen Johnson, in which he makes the point that history is worth telling because it's a story that's true and historically significant. But it helps us to see how things that we come to accept as everyday common facts actually got to be established. Studying the history of neuroscience also helps us remember the giants who were our predecessors and on whose shoulders we stand to see down the road into the future. And of course, we have to recognize that science is a discipline done by human beings, and as human beings, we often get things wrong or we misinterpret the facts that we see. And I'll make these points perhaps time and again. Stephen Johnson makes the point that when something is a truly historical event and change the way in which the future is unfold, we see things that we would not have come to expect, and we have unexpected consequences. So for example, when Gutenberg invented the printing press, he also created a demand for spectacles, because until there was something to read, nobody knew that they were nearsighted. But having caused the creation of spectacles, he also stimulated the production of lenses, and those lenses led to telescopes and microscopes. So the invention of the printing press gave us the historical developments that also drove the science of our neuroscience. Now let me quickly go through a timeline of some of the major events in neuroscience, starting back in the end of the 18th century when Claude Bernard investigated curare. He was the father of experimental medicine, and he did a lot of experiments. One of his experiments was to take a frog nerve muscle preparation, bathe the nerve in curare, and observe that the nerve still worked, but when he bathed the muscle in curare, the muscle did not work. And Claude Bernard found that this process of curare acting on the nerve muscle preparation caused the muscle to be paralyzed. But he interpreted this in the wrong way. He actually interpreted it incorrectly. He thought that curare was disconnecting the flow of vital fluids from the spinal cord into the nerve. The nerves were regarded as hollow tubes because they could see the myelin, but they couldn't see the axoplasm in between. He got it wrong. It wasn't interfering with the flow of vital fluids down the axon. It was interfering with the stimulation of the nerve communicating with the muscle. Now, when we talk about chemical neurotransmission, credit is usually given to this German with a French name, Émile Dubois-Reymond. And what he actually said was that, well, if it isn't electrical transmission, perhaps it's chemical. And for that, he's given credit. But the real hero of the story of chemical neurotransmission has to be John Newport Langley. He was a scientist at Cambridge Physiological Laboratories, and he worked with a lot of substances and tested them on the British smooth muscle stimulus preparations. He had worked with the herb that gave us pilocarpine and observed that when he stimulated certain nerves in the autonomic nervous system, those nerves react to them very similar to what they would do when he put on muscarine. And he observed that this vegetative nervous system somehow could discriminate the chemical signals selectively. Another big player in the system was Ramon Cajal, who used Golgi stain to identify neurons in the central nervous system as individual cells. And his cell theory was quite at odds with what Golgi interpreted, which was the reticular formation. The problem was that Golgi and Cajal didn't communicate with each other, and Cajal only spoke Spanish. When he wrote his papers in Spanish, no one could read them, and he was disappointed that no one read them, so he sent them to the heads of states of the European countries. When they got to the United Kingdom, they were sent to the Royal Society, where they were finally seen by Charles Sherrington, a man whom we'll come to in a moment. But Sherrington was an immediate fan of the cellular theory, and he recognized that Cajal was an important player, and actually he's given credit for inviting Cajal later in the end of the 19th century to come to the Royal Society to talk about his cellular theory. The Langley Laboratory is given credit for naming the autonomic nervous system, and Langley himself experimented with a great deal of things, including the use of nicotine to stimulate the autonomic nerves and to depolarize them and to paralyze them. But that combination of anatomy and pharmacology and physiology, which today we would call neuroscience, Langley worked out the detailed structure of the autonomic nervous system and gave it its name. He also observed that when he took the extract of the adrenal gland and put it on certain smooth muscles, they reacted to those chemical substances in exactly the same way they would to the sympathetic nerves being stimulated. And from this, he advanced the case that there must be a chemical neurotransmitter in the peripheral nervous system. One of his students, Thomas Elliot, was given credit for suggesting, in fact, that smooth muscles reacted to the sympathetic nervous system in the same way that they did for the extracts of the adrenal gland. Although it wasn't exactly correct, it was a good step forward. Then John Langley himself came up with the idea that there must be certain chemical substances on the surface of these different smooth muscles and glands that allowed them to react one way when they were stimulated by a cholinergic-like substance and another way when they were stimulated by an adrenal-like substance, and he called these receptors. He and Paul Ehrlich came up with the concept at the same time, although Ehrlich was given the Nobel Prize for his conception of the receptor system. Then one of the other faculty in the Cambridge laboratory made the observation that stimulating the vagus nerve released something onto its target structures that was very much like muscarine did when they used the extract of muscarine. And from this, we observed the idea that cholinergic and adrenergic nerves populated the parasympathetic and the sympathetic nervous systems. Now we come to yet another student of the Cambridge Physiological Labs, Sir Henry Dale. Henry Dale was a medical student, but when he finished medical school, he wanted to get married, so he took the first job that he was offered in the Wellcome Research Laboratories. And when he got there, he was assigned to use the ergot herb and to figure out what were the substances that were contained in it. And in fact, he did that, and he identified that ergot was able to block sympathetic nerves, and this was the first adrenergic antagonist. But he went on to make several observations after that. Another person who followed him closely at the Cambridge Physiological Labs was Charles Sherrington. Sherrington completed his medical studies and was clinically qualified to practice, but instead he took a trip with two of his classmates to Europe to investigate the cholera epidemic and to understand how vaccines were able to prevent that kind of infectious disease. He eventually spent a whole year with Sir Robert Koch, who won a Nobel Prize, to study the mechanisms by which he could identify which microbes caused which diseases. Eventually, Sherrington returned to England, qualified to practice, but he did not practice, and he became the superintendent of a veterinary hospital where the animals gave him the opportunity to pursue his real interest, which was the pathology of brain diseases in the brain and in the spinal cord. When we get to the early part of the 19th and 20th, the 1910s, we see that Sir Henry Dale was able to identify that synthetic noradrenaline was much more like the sympathetic nerves than the adrenal extracts had been. Of course, he didn't know that depending on which animal species he extracted from the adrenal glands, he got a mixture of epinephrine and norepinephrine. And although norepinephrine was more like the sympathetic nerves than epinephrine was, noradrenaline was thought to be a synthetic substance, and it wasn't known that it was actually made naturally at that time. Then World War II came and research came to a halt. After the war ended, a pharmacologist in Vienna named Otto Loewi was trying to identify a way in which he could prove that the vagus nerve secreted something onto the heart that was the chemical neurotransmitter. One night he had a dream. He woke up from the dream, wrote down what he thought he remembered, and the next morning when he woke up, he remembered that he had had a good dream, but he couldn't remember what the dream actually contained. A few nights later, he had the same dream again, and this time, so as to not take a chance, he got up and went to the laboratory, so he reported later, and did the experiment. And the experiment that he did was to take two frogs' hearts, stimulate one frog, and take the ringer solution that he bathed the first frog's heart in and transfer it physically to the second heart. You can see on the right that when he stimulated the first heart slowed down. And when he took the liquid from the first heart and put it onto the second heart, after a delay, also slowed down. He called this the chemical of the vagus nerve, Vagasstoff, and the Vagasstoff was the first chemical physiological transmitter to actually be proposed. But the problem was that Otto Loewi didn't really understand all of the details of the experiment he had done, and he couldn't repeat his experiment. He took 6 years to repeat his experiment, and meanwhile several people tried to repeat it in a different way. Some took dogs instead of frogs. And what Loewi didn't realize was that by doing it in the springtime when he did, he was working with a strain of frogs that were in winter season mode, so they were largely cholinergic. But by trying to repeat it later in the spring, he couldn't do so because by that time the frogs had adapted to the weather and were now a mixture of sympathetic and parasympathetic. Many people tried to repeat his experiments using blood instead of ringers, not realizing that the Vagasstoff would be destroyed by the enzymes that were in whole blood. But of course, there are no enzymes in ringers, so Otto Loewi could do it, but he couldn't replicate it because he didn't fully understand the nature of what he had done. Then Sir Henry Dale, although not yet a Sir, found that acetylcholine was a chemical that occurred naturally in extracts of the spleen. He also at the same time discovered that histamine, a natural neurotransmitter, could be extracted from the spleen. Two of Dale's colleagues then went on to show that if they denervated the muscle it would react to acetylcholine in a much more sensitive way than if it had been one of the sympathetic muscarinic agents. And so they took this as a clue that the denervated muscle became supersensitive to its neurotransmitter. Now, at this point, World War II was beginning to break out. The Nazis had banned all Jewish scientists from working in German laboratories, and through a quirk of fate, the Rockefeller University agent touring Germany managed to rescue Sir Feldberg because Sir Henry Dale had asked him to be on the lookout for Feldberg because Feldberg had an assay for acetylcholine. Dale wanted him to join his laboratories in London, and he brought him here to do, and within a short time, Feldberg was able to demonstrate that the Vega stuff was very much like acetylcholine and contained acetylcholine, in fact. And at that point, the opportunity to identify the neurotransmitter as acetylcholine in the autonomic nervous system became valid, and they soon found that the acetylcholine could simulate the actions of the cholinergic nerves through the muscles by close intra-arterial injection. Unfortunately, one of the strong physiological antagonists of the idea of chemical neurotransmission was John Eccles. Eccles had been a medical student in Australia, and he interrupted his clinical studies to go work with Charles Sherrington. Now, Sherrington had discovered, amongst many things, the reflex arcs of the spinal cord and the process of inhibition, which was not very well understood from the studies of the autonomic nervous system, but could be seen in a reflex arc as the combination of both an excitatory and an inhibitory message being communicated to different circuits in the spinal cord. Eccles couldn't explain inhibition, but he was very much against acetylcholine being a chemical messenger because in his experiments he had overdosed the muscle with ezerin, and ezerin eliminated the actions of acetylcholine because the nerve became depolarized. So that concept of his, that it had to be electrical because it couldn't have been chemical, fostered his research for many years. The thing he couldn't explain was the inhibition, and we'll come back to that in a moment. But another of his contemporaries was Walter Buchanan, a medical student at Harvard University, who had observed with the early fluoroscope that when the animal became agitated, the intestines slowed down. And from this, he began his studies of the sympathetic nervous system and its target innervations. He had a very complex idea about how epinephrine and sympathin, which is what he called sympathin E and sympathin I, worked. One sympathetic nerves activated, stimulated, whereas other sympathetic nerves, when activated, inhibited the contraction of their smooth muscles. He didn't quite understand that, but he came up with a very complicated hypothesis to explain it, which no one else accepted either. Unfortunately, he didn't understand that norepinephrine and epinephrine were two different neurochemicals. And when Louis and Dale were awarded the Nobel Prize for chemical neurotransmission in the autonomic nervous system in 1936, Cannon was not mentioned at that time because his complicated theory and his persistence and stubbornness in not being flexible about it may have offended certain people in Sweden. World War II stopped a lot of research, but didn't stop research completely. Stephen Kufler, who was working by that time with Eccles in Australia, used the new microelectrode to record from neuromuscular junctions, and he observed that acetylcholine could simulate the action of the skeletal nerve to the muscle. And by close intra-arterial injection, he observed that there was a delay between the stimulation of the nerve and the contraction of the muscle. And that time delay became very critical. Simultaneously, Ulf von Euler, working in Sweden, identified that noradrenaline was actually a natural substance and that it was identical to what Cannon had called his sympathin I. At that point, Sir John Eccles, not yet a Sir again, but working closely in New Zealand, came into contact with the philosopher Karl Popper. Popper had the belief that no scientific theory could be proven, but if you did the right experiment, you might be able to disprove it and falsify the hypothesis. So Eccles came up with a paper that would explain electrically his hypothesis of how the inhibition of Charles Sherrington's reflex arcs actually came to pass. And he defined this in very rigid terms, and then he set about to do the experiment that would prove that electrical transmission was the way in which central inhibition occurred. Unfortunately, the theory predicted one thing, and Eccles observed the other. He falsified his theory. And so he came to say, well, if it isn't electrical, perhaps it is chemical after all. Another person in the timeline of science that we must mention is Jeffrey Harris. Jeffrey Harris was driven by the idea that somehow the reproductive cycles of all animals was driven by the environment, that winter and summer signals could clue the reproductive system. And he couldn't figure out how it was that the pituitary, which was by then thought to be the source of the reproductive hormones stimulating the gonads, could be controlled by something in the environment. And he said it must be the brain that does it. And he set about stimulating the brain and showing that the brain somehow communicated to the pituitary. And we'll come back to that in just a moment. Another important person at this time was Martha Vogt, who was working in London and that identified that if she chopped up a dog's brain and assayed for noradrenaline, that different parts of the dog's brain had different amounts of noradrenaline, that it wasn't just the sympathetic nervous system innervating the blood vessels of the brain, but that noradrenaline had a separate existence in the central nervous system. Julius Axelrod, working at the National Institutes of Mental Health, wanted to study the metabolism of norepinephrine. And he was able to secure tritiated radioactive norepinephrine, and he injected it into the bloodstream and was amazed to find that almost instantaneously it disappeared from the bloodstream. And when he chopped up the body to figure out where the noradrenaline had gone, he found that it was taken up by the nerve cells, sympathetic nerve cells, in the target tissues of the sympathetic nervous system. And from this he discovered the reuptake system. Roger Guillemin and Andrew Shalley were postdoctoral fellows of Hans Selye in Montreal, who was the so-called father of the stress response. And they wanted to understand, like Jeffrey Harris, how it was that the brain could control the secretions of the pituitary, in this case, stimulating the pituitary to stimulate the adrenal cortex. And they began their quest for purification of the pituitary driving hormones that the brain secreted onto them. Their story is much more extensive than I'm able to go into this afternoon, but the book, The Nobel Duo by Nicholas Wade, will tell you about their competition and the many errors that they took along the line. For example, they both believed when they started their quest for these factors that they could not possibly be anything but small molecules, not possibly to be neuropeptides, which they turned out to be. In the mid-70s, Saul Snyder and several other investigators identified that the brain carried specific receptors for opiates, and from that became the quest for the natural endogenous opioid peptide systems. Sir Hans Kosterlitz and John Hughes were given an Lasker Award for their identification of the endogenous peptides called enkephalins. And Avram Goldstein, working at Stanford, identified yet another line of endogenous opioid peptides, which he called the dynorphines. In Japan, in Kyoto, Drs. Numa and Nakanishi changed the entire process by which materials were discovered. They were able to identify from pituitary glands the messenger RNA for the pre-opiomelanocortin gene, the gene that gives rise to both opioid peptides in the pituitary and to the precursor of ACTH. In 1980, Thomas Hochfeld and his colleagues in Stockholm were able to show with immunohistochemistry that the neuropeptides, such as the opioid peptides, occurred in neurons that also contained small molecule neurotransmitters, and that neuropeptides coexisted with those other neurotransmitters. A very revolutionary concept that was very hard for many people to accept at the time. And one of the last elements in this timeline that we'll go into is Dr. Mechoulam and his discovery of the endogenous endocannabinoids that he first called anandamide, and which represent the receptors at which cannabis, marijuana, actually has its effects on the brain. And we take that timeline and reduce it, we can see that there were many people who played important but lesser roles, and several people who played major roles. And these 7 scientists played, in my opinion, the major roles in bringing us to where we are today in neuroscience chemical communication. Sir Charles Sherrington, who discovered reciprocal inhibition in the spinal cord, was a champion of the neuron theory from the beginning. His reciprocal inhibition identified the circuits and predicted that there would be certain circuits that are identical for excitation and identical for inhibition, and that both excitation and inhibition worked at once, that the brain was not at rest waiting for important excitatory signals to come in, but that it was in a dynamic equilibrium and called to action by selectively augmenting inhibition or excitation. Henry Dale was the champion of chemical neurotransmission from the beginning. He identified acetylcholine, he identified oxytocin, histamine, adrenergic agents, and antagonists, but his one shortcoming was that he didn't know that noradrenaline was a natural substance, and so he came close to identifying the autonomic neurotransmitters, but not completely so. John Eccles, the strong opponent of chemical neurotransmission until he falsified his own theory, was perhaps the major driving force for the next half of chemical neurotransmission's history. Because once he had recognized that it must be chemical, he raised questions that drove the science for the next 50 years. What other chemicals besides acetylcholine could be neurotransmitters? How does the actual neurotransmitter substance in the nerve terminal get replenished when it gets depleted by the nerve activity? And how is it that the cell surface of the target tissue of a neuron or the muscle or the gland is able to react? What is it that causes the change in excitability of that target cell, the gland or the smooth muscle or the neuron? His questions drove Peyton to identify certain criteria for identifying what a chemical neurotransmitter must be. It has to be shown that it can be present and made by the presynaptic neuron. It has to have a process that is exactly like the postsynaptic cell reacts to the stimulation of the nerve, and it can reproduce, in pharmacological terms, the transmission from nerve to muscle with agents that simulate or antagonize it. And antagonists have to compete with both the natural substance and any substance that simulates its action. And Peyton said that none of these by themselves will establish that a neurotransmitter is the transmitter for this circuit, but the evidence of all of them put together is what makes the combination the most powerful proof that this is the neurotransmitter. Now we'll continue that with Julius Axelrod and Seymour Ketty, who are major players in the establishment of chemical neurotransmission. They believed in the monoamines, norepinephrine, serotonin, and as you'll hear from Arvid Carlson, dopamine as well. They were able to establish that neurotransmitter transporters, the reuptake systems, conserve the neurotransmitter after it has been released. Dr. Ketty was the first person to propose the biological hypothesis of depression, and he later extended that to mania and to schizophrenia. He was the first person to identify and study blood flow in the brain and show that the oxygen was driven by neural activity, as was cerebral blood flow. And He was the first to ask whether the monoamine transmitters could be the cause of the neurotransmission that led to depression and/or mania. At the present time, scientists believe that chemicals do cause the communication between neurons, that the synaptic activity secretes a chemical agent that transmits the activity from the presynaptic neuron to the postsynaptic neuron, and pharmacologists believe that every new neurotransmitter discovered represents a whole new class of receptors, perhaps multiple classes of receptors, and those mechanisms are opportunities for therapeutic intervention. Medications can be developed to simulate or antagonize neurotransmitters when they are causing disease by deficiency or by excess. And it is that belief that has driven most of my research career. In fact, when I first got to the NIH, I was given the opportunity to work with 5-barrel micropipette electrodes, which is a way to circumvent the blood-brain barrier and apply putative neurotransmitters to the surface of neurons as they existed in the central nervous system. Unfortunately, the results of this early experiment, the pharmacology of individual neurons, was that about 1/3 of the cells would respond with excitation to anything that was spritzed on them, 1/3 would respond by inhibition, which spritzed on them, and 1/3 would respond by not responding at all. And it seemed to me that just asking whether a neuron would respond to a substance was not the key question. The key question was, where is the neurotransmitter presynaptic terminal on this cell, and what chemicals does it contain? And my goal had been to study norepinephrine, and so you'll see here 10 different ways that over the years we have come to develop methods for doing visualization of norepinephrine so we can see its synaptic terminals in the brain. And one of the places that we chose to study this with George Siggins and Barry Hoffer in the early 70s was in the cerebellum, because Sir John Eccles and his colleagues had made that the best understood place in the brain for synaptic transmitter circuitry. And so when we applied norepinephrine, and later on when we stimulated its origins in the locus coeruleus, we found that we could simulate all of the effects of norepinephrine by 5-barrel micropipettes doing agonists. We could block the effects of the locus coeruleus and the effects of applied norepinephrine with beta-adrenergic blockers, and we could extend the effects of norepinephrine through adenylate cyclase by releasing closely cyclic AMP or chemicals which would extend the lifetime of cyclic AMP. And I have, because of the limits on time this afternoon, eliminated all of the G protein coupled receptor discussion, but another major chapter in our history of chemical neurotransmission is represented by that. Now, having identified that norepinephrine could simulate the effects of the locus coeruleus, Gary Aston Jones and Steve Foote studied the locus coeruleus's discharge rate in awake behaving rats and in awake behaving monkeys. And they found that the locus coeruleus would respond rapidly to any novel stimulus in its environment, and that this novel stimulus caused the locus coeruleus to activate its synaptic transmitter systems and circuits all over the central nervous system. When we look at the synapse, we can see that when the locus coeruleus is firing at low rates, it activates alpha-adrenergic receptors, which have a high neuroreceptor affinity, and when they stimulate phasically, they stimulate the beta-adrenergic receptors, which is what we observed in the cerebellum. So the entire system, from the locus coeruleus to its synapses and terminals throughout the central nervous system, gave us an opinion that norepinephrine could be the neurotransmitter for causing focused attention. And Gary Aston Jones and John Cohen have elaborated this in a theory of the norepinephrine adrenergic circuits being attention-gaining circuits. Now let's return to our 7 key players. Jeffrey Harris, driven by the idea of what causes reproductive cycles to be driven by environmental signals. He thought that if he stimulated the brain, and the brain was controlling the pituitary, he might be able to release hormones from the pituitary. So he studied the thyroid system, because with radioactive iodine, he could know when the thyroid gland was active. And he found that when he stimulated the brain, in fact, when he stimulated discrete spots in the hypothalamus, he could drive the secretion from the pituitary of a hormone that would stimulate the thyroid gland. But he said, there are no nerves from the brain to the pituitary. How does it communicate those signals? He was fortunate to come into contact with a Romanian scientist who told him about the pituitary portal system. And so he was able to show the pituitary portal system was a series of blood vessels from the base of the brain from the hypothalamus to the pituitary, and they thought that perhaps through this portal circulation, brain factors could be secreted that would then selectively drive the glands of the anterior pituitary. The posterior pituitary was connected by nerves to the oxytocin and vasopressin neurons in the supraoptic and parasympathetic ganglia, parvocellular ganglia of the hypothalamus. And those stimulations caused them to be secreted. But if there was chemicals released from the brain to drive the anterior pituitary gland, it would be difficult to find those. And so Andrew Shalley and Roger Guillemin, along with Don McCann at Southwestern University, began a quest by going to the butcher houses of Texas, where they were able to get millions and millions of brains and eventually millions and millions of hypothalami, and to use those in extracts with giant Cephedex columns to try to purify these protein factors from the hypothalamus. Eventually they were able to do so, but it took more than 50 tons of sheep hypothalamus to deliver 1 milligram of the thyrotrophin releasing factor, which caused the anterior pituitary to release thyrotropin releasing hormone to stimulate the thyroid gland. The first neuropeptides that were discovered in this way were the tripeptide for thyrotropin releasing factor. Almost the last one to be discovered was discovered by Wiley Vale, the corticotropin releasing factor. Unfortunately, Wiley Vale was lost to neuroscience by his early death. In 1971, I led a session at the Neuroscience Research Program that was designed to ask whether amino acids and polypeptides could possibly be neurotransmitters. We had some of the giants of the neuroscience community attending this conference, and they gave their very strident views. And after a series of discussions, it was concluded that, well, perhaps neuropeptides might be considered to be neurotransmitters, but glutamate and aspartate and GABA couldn't possibly be, because every nerve cell that was responding to glutamate was excited, and every nerve cell that was tested with GABA was inhibited. And these actions were thought too nonspecific. And in fact, it was said, well, how could a neuron possibly keep the glutamate that it uses for protein synthesis separate from the glutamate that it uses for neurotransmission? And again, the neuroscientists of the day were quite wrong. We see in this graph a curve of the growth of the discovery of neuropeptides, largely driven by chemical processes that identified the features of neuropeptides, their C-terminal amidation. I've diffused the names of them so that you can't actually read them, because neuropeptides were given names for where they were discovered or what they did in their first action, but later were found to be all over the brain and to give rise to actions that were not described by the names that they were given originally, such as somatostatin, which inhibited growth hormone when released in the pituitary, but was a brain neurotransmitter everywhere. The last person of the 7 I want to mention is Dr. Numa of Kyoto University. He and his colleague Nakanishi described in this Nature paper in 1979 that by taking the pituitary intermediate glands from 40 different cows, 40 animals only, he was able to identify the messenger RNA that contained the gene that gave rise to the pro-opiomelanocortin hormone. He and Nakanishi later went on to describe the messenger RNAs and the gene structure for the sodium channel and the calcium channel. And by their use of molecular biological techniques, they were able to totally reverse the way in which neuropeptides and other brain-derived factors were being discovered. Instead of looking for proteins to be purified, they were able to identify them completely by isolating their messenger RNAs. And many scientists have used this technique, including my colleague at Scripps, Greg Sutcliffe, to identify many important genes that the brain makes as chemical signals. These are the structures of the first 3 opioid peptide gene families, the pro-opiomelanocortin, discovered by Nakanishi and Numa, the pro-metenkephalin, discovered by Hans Kosterlitz and John Hughes, and the pro-dynorphine, described by Avram Goldstein. But in fact, there are other genes that make opioid peptides as well, although time does not permit me to go into them this afternoon. Thomas Hochfeld described peptide neurons in the autonomic nervous system and then in the brain, in which a neuropeptide was accompanied by GABA or glutamate or norepinephrine or dopamine or serotonin. At first this was very hard to believe, but electron microscopy has later demonstrated that the neuropeptides are occurring in in large granular vesicles and in nerve terminals that also contain small vesicles that contain the more classical small molecule neurotransmitters. This line of research has recently been extended by Maricela Morales at the National Institute of Drug Abuse, who showed that dopamine neurons in a certain select subpopulation can also release either glutamate or GABA, complicating the coexistence property, but making it one of the more compelling questions of neurotransmitters that we face today. So at the beginning of this century, we scientists knew, I put "new" in quotation marks because what we know often is changed by new facts. We knew that there were neurotransmitters that were amino acids, that were monoamines or neuropeptides, but that there were also other chemicals that neurons could use to communicate. For example, there are neurotransmitter-like substances stored in vesicles, such as ATP and adenosine, as discovered by Jeffrey Bernstock. But there were also materials that are formed on demand, such as the prostaglandins and the endocannabinoids, that are not stored in vesicles, and that perhaps even gases like nitric oxide or carbon monoxide or hydrogen sulfide can be used by neurons to signal to the neurons to which they are connected. One of the most compelling examples of these other neurotransmitters is the lactate hypothesis developed by Pierre Magistretti and his colleagues in Lausanne. Magistretti observed that glucose utilization by the brain is sometimes in excess of oxygen utilization. And he had discovered that neurons in certain parts of the brain can undergo aerobic glycolysis, as described by Warburg, in which glucose is metabolized to lactate, and the lactate is converted to pyruvate, which then enters the tricarboxylic acid cycle and can generate 17 molecules of ATP from one molecule of lactate to pyruvate. Whereas when it's just glycolytic metabolism, only 2 molecules of ATP are constructed from each molecule of lactate. He observed that when neurons excite each other, the glutamate that they secrete to excite is taken up by astrocytes, and the astrocytes convert that glutamate to lactate, which they then feed to the neurons as their energy source. In fact, the lactate that glia secrete to signal and provide energy to neurons can be used to control a wide variety of things that include plasticity and all of the genes that underlie plasticity, neural degeneration, and perhaps even the amyloid hypothesis is in part derived from the default circuits in the brain that use aerobic glycolysis as their preferred method of energy production. And it's through this signaling activity drawing glucose and oxygen to the site gives rise to the magnetic resonance imaging methods of functional connectivity imaging that you'll hear in some of the other talks later today and tomorrow. So, through histochemistry, electrophysiology, and pharmacology, we understand neurotransmitter systems. We can identify precisely their anatomic projections. We can identify their behavioral repertoires. But suppose 5 years from now, when David Van Essen's Connectome project reaches its peak, we know all of the neurocircuits in the brain, all of the neurotransmitters, all of their cellular routes, and all of their methods of firing in response to environmental and internal signals. Now that we know that, what is it we know, and how can we build on that to explain how the brain does what I'm doing with you this afternoon, communicating in words thoughts that are complex and creative and hopefully inspiring to you? How can we build upon this knowledge to identify how those abstract properties of brain and behavior emerge from these details of chemical communication and cellular chemical signaling? Now, I began with this talk with a mention of Claude Bernard in the 18th century. Let me take you back even further to the painting of the Sistine Chapel by Michelangelo Buonarroti at the beginning of the 16th century. This is a painting from the ceiling of the Sistine Chapel in which we can see the creation of Adam. In 1990, a physician named Frank Meshberger looked at this painting and said, "Isn't it strange that God's cloak is shaped like the human brain's cortex? And isn't it funny how God's knee is sticking out exactly where the optic chiasm would be? And isn't it funny that that scarf is exactly how the vertebral artery would be?" And he identified that Michelangelo had hidden in his painting of the Sistine Chapel the human brain, by which it is thought Michelangelo meant to communicate that God not only gave Adam life, but he gave him the intelligence of the human being. So I ask you in conclusion, when God touches Adam, was this perhaps the first synapse? And those are my views of the messengers of the mind, and I'd be pleased to answer any questions that may have emerged in the audience. And thank you and good afternoon.
Thank you, Dr. Bloom, for that informative presentation. Your audience will now have the opportunity to ask you some questions. Before we get started on the question and answer session, I would like to remind the audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, is it possible to promote practical neurogenesis in the brains of individuals who are in their 90s, and are there recent publications?
Well, the question of neurogenesis in the brain is an interesting one, and again, it's another example of how initially scientists got it wrong. We thought that as the brain finished development in early infancy, that no new neurons were born, but scientists have subsequently discovered that neurogenesis can be stimulated and that certain stem cells persist in the brain and can continue to generate neurons in certain parts of the brain. And indeed, there are scientists who believe that it will be possible eventually to stimulate these, not so much in the aged brain alone, but in brains as we pass through adulthood, to keep the brains vital and dynamic. It is true that neurogenesis as a natural phenomenon decays with age, but we don't yet have a good way to control that, to do it therapeutically as an intervention.
Okay. Your listeners were engrossed in your presentation, and I'm going to give them a few more moments to ask you some questions. But you know what, we are nearing the end of our session, so I think we're going to wrap up now and close. We are out of time. I'm so sorry.
Unfortunately, well, if they have questions, they can email me and I'll try to answer them.
So I want to thank the audience and for their participation, for attending today's event. And I'd like to thank our speaker, of course, Dr. Bloom, for being here today and for his enlightening presentation. Today's webcast will be available for on-demand viewing for several months following. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.